Hello, welcome. So glad you found it. This is our first video lecture. We're going to try it, see how it goes. This is 13.3 mutations. That's what we're talking about today. As a reminder, these are your class notes, so you should be taking them just as you would in class with a notebook out. The benefit of this is you can stop or pause it to write things down, rewind it. Let us get started. Today we're talking about mutations. Okay, so a mutation is a heritable change in genetic information. What does heritable mean? Heritable is when you can pass things down from generation to generation. So how parents pass down genes to their child. In this case, a mutation is a change in the genetics that is passed down. There are two types of cells in your body. You have body cells, which are specific to you, that don't get passed down. And then you have sex cells, which will get passed down to your children. Mutations can only be heritable, passed down, if they're in sex cells. That's important to remember. This is a picture of a man who lives in Indonesia. They call him the tree man, and here's why. Because of the human papillomavirus that this man unfortunately has, it has changed his genes that code for his skin. His skin develops this very bark-like quality, like a tree. It's a very rare disease, but it is one that is very extreme, and you can visualize it here. Let's talk about the first of two types of mutations. This one is called a gene mutation. Okay, a gene mutation happens when you have a change in just one or a few handfuls of nucleotides. Remember, nucleotides are made of three things: a sugar, a phosphate, and a base, just as review. Our first type of mutation is point mutations. It's a change at a single area of the DNA, a single point. It's very easy to think about that. Now, on this example, it says substation. I meant to say substitution, so if you could change this word to substitution, that would be great. The next slide will show you what substitution is. It's when you take a single base and change it out for a different base. I'll show you a picture in a second. The second type of a gene mutation is called a frame shift. Now, we talked about on Tuesday that codons is how you read code. Codons is 3 bases at a time. That's how we look at code. 3 bases together will code for an amino acid. We know amino acids chained together will make a protein. A frame shift mutation occurs when this reading frame, so we read things in 3 bases at a time, is shifted. So it shifts the message. So if we had A, U, G, and bases on either side, if we had a frame shift mutation, we'll shift it in a way that we won't read AUG anymore. Okay? We'll look at that in a second as well. The example would be insertion and deletion. Let's take a look at the picture. So our first one here is substitution. Okay? As you can see, here's the original strand. In this case, we have a G. When we go under substitution, what we get in the next strand is that base is now no longer a G. In this case, it's a C. What we've done is substituted one base for another. Let's look at insertion, our second one. Okay, so we're back to this strand right here. When we're inserting, we add a base. So we had a C and an A right here, but now on our new one, we've put a G right in between them. Insertion. The last one is deletion. As you can imagine, if we have this, we start with this strand, we have this T right here. When we undergo deletion, we no longer have the T. So you can probably figure out from their names what that means. Okay, I'm going to erase this and show you what a frame shift mutation might look like. So, to review, we talk about genetic code and codons. So for example, that's a codon. It's 3 bases put together. Okay? If we have a frame shift mutation, the codon might shift to here. See, our frame has actually shifted. It's a mutation that has caused the bases to change in such a way that we no longer have CGA, but now it's shifted and we have TCG. Okay? It's a frame shift. Our second type of mutation is called chromosomal mutations. What happens in chromosomal mutations is that it's a change in a number or structure of the chromosome. Chromosome is the bigger entity of genes. We're not talking about single base, but we're talking about an entire chromosome at a time. Changes in an entire chromosome are more detrimental. It affects the individual, it affects the organism at a greater level. Sometimes gene mutations, like point mutations, don't result in any change. You might not notice a change in any protein, but a chromosomal mutation are definitely more serious. We're going to talk about the 4 types. I'm going to show you a picture about what they are. They are deletion, duplication, inversion, and translocation. So if we look here at the first one, deletion, on the left we've assigned these random areas on what you see is a chromosome, letters, so that we can track them. So we're starting here on this side with the D. After deletion occurs, we no longer have the D. It goes from B to C. Okay, that's a chromosomal deletion. This entire portion of the gene of the chromosome is deleted. Okay, here's our second one, duplication. We start with B and C being together. After it's duplicated, now we have 2 sets of BC, BC. So we've added a large portion to the gene. Inversion is the third one. So right here we have BCD in the right order, but when we invert it, we've flipped it. Now we have just the opposite, DCB. Our fourth one is called translocation. In this case, we're looking at 2 genes at a time. So we have a gene here, we have a gene here. We have the areas highlighted, AB on one and MNO on the other. After translocation happens, what's actually occurring is this block is popping off of this gene and this block is popping off of this gene, and they're switching. Okay, so now on the other side, we see MNO on the top one and AB on the bottom. If you look at the letters on the original on the left side were in order, but now they're not in alphabetical order, okay? Translocation has occurred. These chunks have broken off and translocated to a different part of the gene. So now we're going to move on to our second topic, and that's effects of mutation. What effects do mutations have? So, mutations can be caused by a natural event. It can be caused by an artificial or man-made event. There's lots of different things that occur. It can occur by an environment. It can occur just by chance, by nature. It can occur by an effect of nurture. There are many things that can cause a mutation. And it's important to remember that a mutation may or may not cause harm. It doesn't have to cause harm. Like we said, point mutations a lot of the time will change one base, and that won't make a difference in the coding of amino acids. Because if you remember our reading codons circle, our decoding circle, oftentimes the third base in a codon could be any of the 4 bases, and it'll still code for the same amino acid. If you don't know what I'm talking about, refer back to that reading codons or decoding codons circle we've used. Mutations can also be from errors in the genetic processes. So when we looked at replication, we saw that DNA polymerase added bases and made the hydrogen bonds between the new bases so that we can make a new strand. But the second job of it that we talked about was that it proofreads. If that proofreading ability gets lost, it gets damaged in the DNA polymerase, it can cause a mutation, which means if one of the bases is paired incorrectly, but that proofreading ability is no longer there, then it can cause that gene to be permanently wrong. The bases could be permanently paired up in the incorrect manner, an incorrect replication. This term you need to know is called mutagen. A mutagen is just a chemical or physical agent in the environment. So mutations can often be caused by mutagens. Examples would be smoke. That's why Fishers has gone no smoking, because smoke is harmful to your health. It causes mutations. Many people who smoke get lung cancer. This is because the chemicals in the smoke after a period of time will build up and cause your genes to change. Change in genes can cause any number of diseases, including cancer. Radiation is another big one. That's why X-rays are good in very small quantities when you need them, but if you have X-rays too often, it can actually lead to cancer. The way that this happens is the X-rays degrade your genes in which they're passing through. It changes the genetic code, and it can often result in cancer. Any of these mutagens in a high frequency will cause rates of mutation to increase. There are two types. You can have harmful effects, and believe it or not, you can have beneficial effects from mutations. Harmful effects could include, the most dramatic of them would be when you've changed to the entire function or the entire structure of the gene or the chromosome. Okay? Defective proteins then, from these harmful effects, right? So we've changed the genes, we've changed the bases. We know when we do that, we affect the amino acids that they're then coded for. They're coding for different amino acids, which means it changes the protein. That the ultimate protein we were looking for the end is now different. So when you have a different protein, it can't function in your cell correctly or can't function in your tissue, whatever we're talking about at the time, and it changes the biological function. An example would be sickle cell anemia. It has to do with red blood cells. So if we see on the left, here's a normal red blood cell, is looking nice and circular. When we have a normal red blood cell, it passes through our veins, our arteries, and it's unrestricted because of the circular quality. If you have a mutation to the hemoglobin molecule, hemoglobin is found in red blood cells. It's what carries the iron. If you have a mutation to the hemoglobin portion of your red blood cell, it can result in a sickled shape, which is what this is. If your blood cells have a sickled shape, A, it's harder for oxygen to attach to them, and B, when they move through your body, the sickle cells block the blood flow. Because they're not circular, they can build up in this clump right here. A clump in blood in your artery moving throughout your body is not good because you need blood to get to all parts of your body. This can result in very serious health issues. We have 3 more slides to go. Let's talk about the beneficial effects. So beneficial effects of mutations, it can be useful to organisms, believe it or not. So we use pesticides on our foods and fields. It's a very common thing to use in farming. It helps get rid of the insects so that the crops are better, they're better for us to eat, they're not rotten, they're not holes in them. But some mutations in insects have caused them to be resistant to pesticides over time. So for humans, that's not good, it's ruining our crops. But when we look at it from the perspective of the insects, this type of mutation, this resistance is good for them. It allows them to eat, have more food. But humans are also finding that sometimes we're growing resistances to certain diseases, like HIV. These are good things. There's a term called polyploidy. I want you to know that. That's when an organism, because of mutation, gains an extra set of chromosomes. This can be beneficial in some ways. If we talk about plants that exhibit polyploidy, often are larger and more hardy. So farmers are selecting for plants that have an extra set of chromosomes. It can result in this. This is what we would call a normal strawberry. These are polyploidy strawberries. These are like the most Hulk-like strawberries that I've ever seen. This picture was obviously not taken in the US. This is a different currency, but the example still stands. Polyploidy results in larger, more hardy crops. We have made it. You've made it through the first video lecture. I hope it went well. Ultimately, it will go faster than doing it in class, and we will have more time to use in class. So, before you're done, I would like you to take my quick 3-question Nearpod quiz, and you can access it via this code right here. So if you go to nearpod.com or go to your Nearpod app, type in this code, SNGLM, and that should take you to my 3-question quiz. If you have any issues getting on the quiz, you need to email me before the next class. Starting at the next class, I'm going to download the results from the Nearpod quiz and enter them into the gradebook, okay? So if you have issues, you need to let me know before class. It has been great. I hope you enjoyed this, and we will see you soon.